I'm a postdoc with Giles, and I told him not to vote for me, so it's not even a rigged competition, I promise. So the hypothalamus is a small region of the brain in the center, kind of bottom edge of the brain. And this position puts it in a very unique place where the blood-brain barrier isn't as strong of a barrier. So the blood-brain barrier in this region is a little more permeable, and this allows things that are circulating in your blood to reach this part of the brain. So some of these things that could be circulating are metabolic hormones like insulin, leptin, or ghrelin. It can also sense other things like nutrients in your blood, something like glucose. So all these signals are coming from your body, and they're being communicated to cells in the hypothalamus. And then the cells, mostly the neurons in the hypothalamus, are able to send on their messages to then regulate your appetite. But of course, appetite regulation isn't always happening perfectly. If your appetite is too large, you could be overeating, leading to increased body weight and a higher body mass index value. Or to the other end of the extreme, you could have a smaller appetite than needed, so you're not consuming enough calories and you could have a very lean, low BMI. So the purpose of my project is to really investigate what could be happening in the hypothalamus cells of these patients to see if we can better understand the cells and the genes that could be functioning behind this appetite dysregulation. So one of the tools that I'm going to be using for mapping the hypothalamus is single nucleus RNA-seq. So here we get frozen brain samples from these patients, and then we immediately just chop it up into little pieces and extract the nuclei and then sequence the genes that are being expressed within each nucleus. This is cool because we can process a lot of nuclei at once and get a lot of information about the whole genome transcriptome. But the downside is we immediately chop up the tissue, and sometimes the organization of cells within a tissue can be very important for their function and just be information that we don't really know. And so there's the downside to this where we are losing all of that 3D organization information. So to supplement that, we also do something called spatial transcriptomics. And this comes in two flavors. The first one is standard definition, where we have a thin slice of tissue and we lay it against a glass side that has large barcoded spots. And so all of these barcodes help us remember where these cells used to live within the tissue. So when we're measuring the gene expression, similar to sequencing, we're still remembering where it used to live in the tissue. That gives us more information. This is a technique that allows us to measure a larger area of tissue, but the resolution's pretty poor. We're not really seeing a single cell resolution. We're kind of seeing an average of maybe a few cells in a region and what those genes being expressed there were. So to help this, we also have a high definition version of this protocol where the glass slide is now fancier and smaller, but we have much smaller spots and barcodes. So instead of getting an average of a few cells and their genes, we can now see a single cell or maybe even a subcellular region and measure gene expression there. So our design is to take samples from a variety of people at different extremes of the BMI scale, and we will chop their tissue into coronal sections so we can get a picture of the entire hypothalamus front to back, and then perform all of these techniques. Once we get our data back, we'll start integrating all the data sets together. We'll be able to use the spatial information from the spatial transcriptomics techniques and use that to infer where the single nucleus data set used to come from in the tissue. And we can use the large number of nuclei we're characterizing from the single nucleus RNA-seq to supplement the smaller sample size we get from the spatial data. And together, all of this will help us build a 3D space where we see cells in the hypothalamus and which genes they used to express. And this is something we've done before as a lab in both humans and mice, but the novelty of my project is we're having samples from people with extremely low or extremely high BMI. So this will give us the first opportunity to really characterize what cellular differences are between these people. Maybe there are different cell types more or less present, maybe they're in a different area. And then even below that level, at the gene level, which genes are being expressed in these cell types to really get close to understanding how this is happening on the molecular level. So the ultimate goal is we'll better understand just the normal human brain, but also brain expression patterns at extreme weights, and maybe we'll be able to better understand how our current obesity drugs are working and maybe discover some new ones that work even better. Thanks.